Hello, everyone. This is Dr. Hall. Welcome to Lecture 5, which is all about synapses. I've broken this lecture down into 5 different parts. First, we're going to do a quick review of action potentials because it's relevant. Then we're going to talk about the 2 main types of synapses. And then the last 3 sections all about neurotransmitters, how they're released, how they do their actions, and what happens to them is true of chemical synapses. So let's get started. So a quick review of action potentials. A neuron has to decide to send an action potential, and you may recall that it is going to be the axon hillock that is the decider. So what's happening is that neurons are constantly receiving signals from other neurons. Those are generally neurotransmitters that cause ion channels to open. And if that ion channel allows an ion to move in a way that depolarizes the cell, brings its resting potential, membrane potential from -70 closer up to -55, then that's going to be excitatory, moving you closer to threshold. If that ion channel opening allows charged particles, ions, to move such a way that moves you further away from that -55, then that's an inhibitory postsynaptic potential. So all of these get added up. You should remember, right? So you're getting these different signals, they're all going to add up, and if you get to that -55 for your membrane potential, then the axon hillock is going to open sodium channels. Sodium's going to rush in, and we're going to go all the way down the axon. So remember, summation of all of the different signals, all of the different changes in membrane potential that are happening due to various inputs. Oh, there's that -55. Once we reach that -55, boom, action potential. So the phases of the action potential. So the sodium channels open, and because of its electrochemical gradient, sodium ions are going to rush into the cell. Sodium ions are positively charged, so the cell becomes less negative. I don't know, can you see that yellow? I don't know, let me try the highlighter. Okay, so we have this rising phase here. Then the sodium channels become inactivated, potassium channels are open, and because of its electrochemical gradient, potassium rushes out of the cell. So then the inside of the cell becomes more negative, and we have this falling phase. Then everybody closes, and the sodium-potassium ATP pump puts things back to where we were. So that is what's happening along each segment of the axon, boom, 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 all in a row, down the length of the axon. So here's this little animation showing that, right? So the action potential, you'll recall, only goes in one direction. That is because of the absolute refractory period, the relative refractory period, right? It can only move in one direction, and it's an all or nothing event. Either it happens or it doesn't, and then we're going to end up down at the axon terminal. All right, so now we have arrived at the synapse. So we're going to talk about the different types of synapses and their anatomy and kind of how they function. The definition of a synapse is it's a place where one neuron is communicating with another neuron. So it's the place where you're signaling from one cell to another. So the cell that is sending the signal, the cell that is talking to the other cell, is called the presynaptic cell because it's the one before the synapse. And then the one that it's talking to, that's on the other side of the synapse, is the postsynaptic cell, right? Because pre means before and post means after. And then the synapse is the place where these two neurons are in some sort of communication with each other. So the 2 main types of synapses that we have are electrical and chemical. If it's electrical, it's all about the movement of charged particles, because that's what electricity is. In this case, it's going to be ions. Those are the charged particles that are moving in our cells. If it's chemical, then we're using molecules to send that communication across the synapse, and we call those molecules neurotransmitters. So neurotransmitter is just the fancy name that we use to call these different types of molecules that act as transmitters of a signal from one neuron to another. So we have electrical and chemical. Electrical synapses are really interesting. So you have two cells that are next to each other. So let's say here's my two plasma membranes, and they're not, let's see, there we go. They're not directly touching each other. There's a little space. But for an electrical synapse, you have these special proteins that form these little channels or like tunnels called connexons. So you can kind of see it over here. So it forms these little tunnels, these little channels, where the inside of one cell is in direct communication with the inside of another cell, and things can flow freely to them. So they're two separate cells, but yet there's this place where they're in direct continuity with each other, really. It's kind of like if you've ever stayed at a hotel where you had adjoining rooms with somebody else's room, right? So like you had a door you could open and then you can walk back and forth freely between the two rooms. It's kind of like that. Each cell is its own cell. It has its own set of all the organelles and all that kind of stuff, but they have this direct communication link between each other through these connexons. Now, the one difference between this metaphor here of the, or simile, I guess it would be, of the adjoining hotel rooms is that in this situation, there's no door to close, right? There's just always this open communication. So what happens in electrical synapses is that if one neuron has an action potential, goes down to its axon terminal, and then it's at an electrical synapse with another neuron, it just goes right through, right? Those sodium ions rushing in are going to rush right into the postsynaptic cell, and right, those sodium ions rushing in is what opens the voltage-gated sodium channels, and so that action potential is just going to propagate down that next cell, and then if there's another cell in an electrical synapse, and on and on and on. So electrical synapses are really interesting and really powerful. They are super fast. Okay, this happens just as fast as an action potential down the axon. The action potential then moves into the next cell. So it's super, super fast. It's basically instantaneous, right? We're talking like micro thousandths of a second. And it allows you can have a whole string of neurons that are all connected by electrical synapses, and they're basically going to act as a unit. So if one of them gets triggered to fire, then they basically all go boom, right? So that's kind of cool. It's also bidirectional. So because they're in direct communication from each other, if this one does an action potential first, that'll send the next one. But if this one sends an action potential first, that'll send the one next to it. So it can go in any direction. So it's pretty cool. They're pretty awesome. So there they are, an interconnected network. Here's an example of how, things can go either way. So it's bidirectional. But it doesn't have a lot of finesse, right? Either they all go or none of them go. There's no such thing as kind of a strength of anything. There's no strength of signal. It's just like a big yes or a nothing. And so we don't use these a lot in human nervous systems. We do in a few places, but we don't use them a lot overall because they're hello, Oliver. Because, you don't get a lot of specific control, and we like to have a lot of specific control. That's where chemical synapses come in. So in this case, the cells don't have any direct connection between each other. There's this space in between them. There was a space between the other ones, but those little tunnels, those connexons, bridged it. And it's called the synaptic cleft. And the space is a little bit bigger than what we saw in cells that had electrical synapses. And the axon terminal is filled with these little vesicles, these little membrane-bound packets that are filled with those neurotransmitter molecules. So that's the structure. And what's going to happen is in order for this presynaptic cell to send its message, it's going to release these neurotransmitters out into the synapse. They're going to drift across and bind to special receptors that are on the postsynaptic cell. Now, depending on what happens when they bind, that could end up triggering EPSPs or IPSPs, right? And all those things that might make a neuron more or less likely to fire. And as you'll see, it can also do some other things as well. So chemical synapses are pretty awesome because you can send different types of signals. It's not just a big go. You can make a neuron more likely to fire, less likely to fire. You can also make it a lot more likely to fire a little bit more by releasing more or less neurotransmitters. So you get a lot of specificity with chemical synapses, which is pretty awesome. They are a little bit slower, obviously, than electrical synapses because those are practically instantaneous. These are still really fast, but a little bit slower. And of course, they're not as in sync, where you can't have a whole network that kind of operates all as one. It's just from one to the other, to the next, to the next, to the next. So if you have a big sequence of neurons that need to kind of communicate one to the other, to the other, to the other, to the other, if you have a long chain, it's going to take longer, right? But that's a sacrifice that's worth it so that you get the finesse. So in summary, electrical synapses have these gap junctions, direct connections made by those connexons that make those little tunnels, those little connections between them. It can be a huge network. As soon as one of them goes, they all go. It doesn't matter which one goes first. They're all going to go. It can go this way, it can go that way, and it's super fast. Chemical synapses are called chemical synapses because we use molecules called neurotransmitters to send the signal. They can only go one way, from the presynaptic to the postsynaptic cell, and they're a little bit slower, but they give us a lot more specificity, a lot more finesse, a lot more control over what's happening. So in the rest of Lecture 5, we'll talk more about chemical synapses and how they operate.